Hello, welcome to 13 Universal Learning Beyond Barriers. Okay, so this is biology class with schedule, and we're going to be looking at A-level Caie biology as usual. The questions that we'll be solving they are all from A-level past questions, Caie. Okay, so we are going into biological molecule series 4. Last time we stopped at series 3. Sorry for any hitches you encountered towards the end of series 3. Okay, however, we're going to sort out everything in series 4. So the first question here says, which explains why hemoglobin is able to carry oxygen? Which explained? Okay, A says it consists of 4 polypeptides. Is that why? B says it contains iron. Is that why? C says it has a quaternary structure. Is that why? And this says it has a global protein. It is a global protein. Is that why? Pause the video and try it. Okay, pause the video and try it. And if you check, you see that why hemoglobin is able to carry oxygen, right? Now hemoglobin is a respiratory pigment, oxygen transport pigment. Don't forget that hemoglobin is a protein, but it has prosthetic groups. Okay, so it has prosthetic group. The prosthetic group is called hem group. Okay, the hem group. The hem group contains iron 2 ion. So now this hem group is a prosthetic group. A prosthetic group is a non-protein part of a protein. Non-protein part of a protein that helps the protein to carry out its function. Non-protein part, right? So it contains iron 2 ion. So that helps hemoglobin to bind to oxygen. Of course, so look at it. It consists of 4 polypeptides. This is quaternary structure. It shows the quaternary structure, which consists of 2 beta globin polypeptides and 2 alpha globin polypeptides. That's not the reason why it's able to bind to this. It contains iron, and that's it. Iron is found in hem. Okay, right, hem group. Okay, so B is likely the answer, but let's look at others. It has a quaternary structure. Quaternary structure, yes, that's 4 polypeptide. That's not the reason why it's able to bind to oxygen or carry oxygen. It is a global protein. Well, that gives it its shape, all right, and it helps it to fold it helps it to fold in ball-like shape that makes it soluble and metabolically active. But for carrying oxygen, I think it should be iron 2 ion. So the answer is B. Okay, let's go to the next question. It says, which shows the basic unit of cellulose? Yeah, I think we should know this. Now the basic unit of cellulose, what do you think? Pause the video and check. The basic unit of cellulose, the monomer of cellulose, that's what they're asking us. All right, so the monomer of cellulose, you know the monomer. Okay, the monomer is alpha beta glucose, not alpha. Okay, beta glucose, right? So how do we know beta glucose? All right, now of course this is a glucose. This is first carbon atom, second, third, right? That is fourth. This is fifth. That is sixth. So this is a glucose, hexose sugar, glucose. And if you check, when I draw a plane of ring across the ring structure, the OH attached to the first carbon atom is above the ring. All right, and that's how we know beta glucose. The OH attached to the first carbon atom is usually above the ring, the plane of the ring structure. So this is likely the answer. Why is B not considered? All right, now look at that. This 1 2 3. If you check this is similar to this, but the challenge here is that, or the difference there is that well, not a challenge, is that if you check, this is the first carbon atom. It's the first carbon atom that differentiates them. All right, the OH attached to the first carbon atom is below the plane of this ring structure. Okay, so because of that, this is not beta glucose, this is alpha glucose. Do you get alpha glucose? Yeah, and that's alpha glucose, right? The monomer of cellulose is not alpha glucose. If you check this one too quickly, 1 2 3 4 5, this is not the arrangement of glucose, carbon to carbon arrangement. If you check this one, this is not the arrangement. It should be 1 2 3 4 5, and then the 6 carbon atom is here. Now since we are dealing with cellulose, the monomer is beta glucose, which I think is A. All right, that's A. Let's go to question 3, which explains why hemoglobin is suitable as an oxygen carrier. This question is repeating again. Why is it suitable as an oxygen carrier? I want you to think about it and solve this question. All right, so number 1 says the hem group combines with oxygen. Okay, number 2 says the ion combines with oxygen, reversible. Yep. 3 says hem group changes shape as oxygen loads. Okay, which one is the answer? Is it A, C, or D? Look at this. A says the hem group combines with oxygen. Yes, that's correct. The M group contains ion 2 ion, and that ion 2 ion helps hemoglobin to combine with oxygen, right? So we have 4 hem groups, and each one combines with a molecule of oxygen, so this is correct. Number 2 says the ion combines with oxygen, reversible. Yes, ion combines with oxygen, reversible. This is very important. This is why hemoglobin is an efficient respiratory pigment. All right, so when hemoglobin, you remember this hemoglobin, this is Hb, all right, hemoglobin, all right, combines with one hemoglobin molecule combines with 4 oxygen molecule, because each molecule combines with a hem group. Okay, and we have 4 hem groups in hemoglobin. So when this happens, the forward reaction, we have this compound. Now tell me, what's the name of this HbO8? So what is it? Okay, this is oxyhemoglobin. This is oxyhemoglobin. This one, oxyhemoglobin. So this happens in the alveoli, okay, where oxygen combines with hemoglobin. But when it gets to respiring tissue, the backward reaction will occur because oxygen is needed. So what we're going to have here is that this oxyhemoglobin would associate to give us hemoglobin and oxygen. Oxygen will diffuse into the tissues for aerobic respiration, while hemoglobin picks up hydrogen ion. All right, so it's reversible. That's correct. Hemoglobin changes shape as oxygen loads. Yeah, this is correct. That's how hemoglobin behaves. If you look at hemoglobin dissociation curve, you find that the curve is a sigmoid shape, right? This part where it's just like this. Okay, so this is partial pressure of oxygen on the x-axis. Y-axis is percentage saturation of hemoglobin. All right, so at this first point, you can see the steep. The beginning is steep, the end is steep. What is happening here is that as hemoglobin binds, oxygen binds to the first hemoglobin hem group, it takes time for that to happen. But once it binds to the first hem group, the hemoglobin molecule will change its shape. Right? It changes its shape to allow, of course, changing of shape allows more oxygen to bind, all right, to the hemoglobin. And that's why we have a steep curve here. All right. So I think that what do you think is the answer? All of them are very important. All of them are very important. Okay, so let's go to the next question. Which diagram shows part of a structural polysaccharide? Yeah, structural polysaccharide is cellulose. Remember that one thing about cellulose is that there's rotation. All right, the adjacent glucose molecules rotate by 180 degrees, so there's no rotation here. All right, this is oxygen. Okay, there's no rotation. It cannot be A. A looks like, right, it looks like amylose. Amylose has a straight chain. Okay, now look at B. Okay, now this glucose molecule, this is it, and then the next one, adjacent one, all right, flips for through 180 degrees. The next one is okay, the second one. So there is rotation of adjacent glucose molecule by 180 degrees. All right, and of course you can see the glycosidic bond also rotating. All right, so this is likely cellulose. Cellulose is a structural polysaccharide. Okay, let's look at this. This doesn't look like cellulose. This is branched. This looks like maybe amylopectin or glycogen because they are branched. If you look at this, okay, it doesn't look like cellulose because cellulose is made up of, this is made up of two different, okay, monomers. All right, cellulose is made up of, which is polysaccharide, structural polysaccharide, because it provides support to the cell wall. Okay, so it's only made up of beta glucose, which is this. And how do we know? There's always rotation. So that's the answer is B, of course. All right, please don't forget to send your comments, share the video, like the video. All right, and then subscribe for the next one. All right, so the diagram shows the structure of two amino acids, each of which has more than one amine group. Now this is what I was trying to say in series 3, okay, towards the end. So a peptide bond is formed between two amino acids. All right, let's see which groups form the peptide bond. All right, okay, which one? Now look at this. I told you guys that if it's an amino acid have terminal carboxyl group and is always linked to the terminal amine group by a carbon. This one, the terminal amine group is this. How do I know? It is linked to the terminal carboxyl group by this carbon. Now this is not terminal because it's not linked to another carboxyl group by a carbon atom. So this falls within R group. The same thing, this falls within R group, and peptide bond is not formed between R groups of adjacent amino acid. It's formed between the terminal amine group of, or terminal carboxyl group of an amino acid and the terminal amine group of an adjacent amino acid. That's the way. So 1 and 3, right? 1 and 3. It cannot be 1 and 4 because carboxyl group cannot react. So this is 1 and terminal carboxyl group react to terminal amine group. Okay, that's 1 and 3, and that is A. All right, that is A. Okay, next question. Which combinations of bond types correctly shows the weak and strong bonds that hold a molecule of protein in shape? Okay, let's see. All right, look at this. Hydrogen bond is weak. Of course, the intermolecular bond is weak. Disulfide is a covalent bond. It's very strong. So it's either C or D. Ionic bond is also weak. It's not an intermolecular bond. It's weak. So hydrogen bonds are broken by change in temperature and change in pH. Ionic bond is broken by only change in pH. Disulfide bond can only be broken by a reducing agent. It's very strong. Okay, so please don't forget that it is D. All right. A student carried out 4 tests for biological molecules on a solution. The results are shown in the table. These are the results. Okay. Yep. So you can see they say which molecules are present in the solution. So look at this, guys. When they did iodine, it remained orange. So iodine is used to test for starch. Okay, so starch is not present. Burette is used to test for amino and protein. Okay, all right, and it remained blue. If it was, if for example, if iodine, if starch was there, it would have been blue black. If protein was present, it would have turned purple or lilac or mauve. Now, Benedict solution is used to test for reducing sugars. All right, and it changed orange. That means it's okay present. Emulsion is used to test for lipid. If lipid was present, it would show cloudy solution while precipitate. So that means lipid is not there, protein is not there, starch is not there. It's the only one is there is blue is reducing sugar. Okay, so if you come here now, this is made up of amino acid joined together, 3 amino acids joined together. Okay, so this is what burette. It shouldn't be there because the question says which molecules are present in the solution is only reducing sugars. Okay, look at it. This is a reducing sugar. This is, what molecule is this? Tell me. I want to hear you. What type of glucose is this? Oh, say it. Okay, yeah, you got it. So this is beta glucose, because the OH group attached to this first carbon atom is above the ring, and this is alpha OH attached to the first carbon atom is below. So both of them are reducing sugar. So so the answer should be what? Is it A or B or C or D? What do you choose? Of course, 2 and 3. The answer is C. Is that okay? All right. So which bond will be broken when a molecule of amylose is hydrolyzed? Alpha 1,4, beta 1,4, alpha 1,6, beta 1,6. Okay, which one do you think is the answer? Look at this. Now, amylose, please, you have to remember that amylose is unbranched, straight chain, polysaccharide. And amylose is a polysaccharide of alpha glucose. So it can never be 2 nor 4 because they are talking about beta, right? Now 1 or 3 likely, but let's check. Because amylose is unbranched, it forms only 1,4. 1,4 glycosidic bond is for unbranched or straight chain. 1,6 is for branched. So it cannot have 1,6 because it's not branched. It only have 1,4 because it is unbranched, right? So the answer is C. What do you think? Right? Okay, let's go to the next question. So which statement describes how the molecular structure of starch is suited for its function? Okay, let's pause this video and check it. All right, look at this. The first one says amylose has a branch. Now amylose is not branched, so this disqualifies this. It's unbranched, right? The second one says in the breakdown of amylose and amylopectin, many condensation reaction. Breakdown is not condensation, it's hydrolysis. Breakdown is hydrolysis, not condensation. That eliminates B. C says in the formation of amylose and amylopectin, many hydrolysis reactions allow now formation of this is condensation. Okay, alpha glucose molecules linking up, all right, joined by glycosidic bonds to form amylase and amylopectin, and that is condensation, not hydrolysis. Formation of polysaccharides is by condensation, not hydrolysis. That eliminates C. This is the amylose amylopectin complex. Amylose amylopectin is insoluble. Yes, starch is insoluble, and therefore it does not affect the water potential of the cell. Do you understand? So starch is insoluble. So that means that starch does not dissolve in water. So water does not move into the cell or move out of the cell because of starch. You understand? All right. So because starch is insoluble, that's why it can act as a storage molecule. Is a store of glucose in plants. So the answer is what? D. Thank you. Let's go to the next question. Which comparative statements concerning biological molecules are correct? That's number 1, number 2, number 3. So which one do you think? A, B, C, or D? Look at it. There's a collagen molecule is a fiber. Yeah, this is correct. It's a fibrous protein, structural protein that contains many amino acids with hydrophobic arrow group. With hydrophobic arrow groups. Okay. Whereas hemoglobin molecule is a globular protein with no amino acid with hydrophobic. No. Some amino acid in hemoglobin are hydrophobic. That is why hemoglobin has globular shape. Why does it have globular? What is globular? Globular proteins are proteins that are folded into, okay, ball-like shape so that the hydrophilic arrow group can be on the surface and hydrophobic can be at the center. Now hydrophobic, so it has hydrophobic amino acid because they are the ones that have hydrophobic arrow groups to give it comparative balancing. Okay, so it doesn't have only hydrophilic. So this statement that says that, you see that statement there, that hemoglobin with no amino acid that hydrophobic, that's wrong. So this is disqualified, eliminated. This is number 1. Then number 2 says sucrose hydrolysis results in bond breakage. Yes, that's glycosidic bond. Production of equal proportion of fructose and alpha glucose molecule, whereas cellulose hydrolysis results in only in beta glucose. Yes, see, cellulose, right, the monomer of cellulose is beta glucose. This is correct. All right, And then sucrose is a disaccharide, right? It contains two monomers, fructose and alpha glucose. All right. So when you break down glucose, you have equal proportion of two of them. So this number 2 is correct. 3 says the glycosidic bond of glycogen have been formed between two alpha glucose molecules. Okay, whereas with aminopectin, the bonds have been formed between alpha glucose molecule and the beta. No, there's no beta glucose molecule in aminopectin. Okay, it only has only alpha. Okay, so the answer is only number 2 that's correct. So the answer is not C. C is the correct answer. C is the only correct answer. All right, so please, this is where we stop in series 4. We're going to series I think we're going to be going to series 5, or we'll go to enzymes. Get ready for enzymes. All right, the video will be coming out soon. Right, please like our video, share it, make comments, okay, and subscribe. All right, so thank you for joining. See you next. Bye. Okay, bye.